Hi everyone, and welcome to lesson 5 of B3, which is plant diseases and defence. Again, this is a triple only content lesson, so if you are a combined student, you do not need to complete this lesson. As always, you will need a pen and some paper, so if you do not have them with you, please pause the video and go and find them now. So today's challenge is to describe how plants defend themselves against disease, and today's Aspire is to explain how certain diseases and deficiencies can affect a plant. So today's starter, I would like you to write down what you think a pathogen is, and I would like you to list the 4 types of pathogens. So I'm going to give you 1 minute to do that. Off you go. Okay, you guys, so you should have written down that a pathogen is a microorganism that causes a disease, and the 4 types of pathogens that you should have written down are virus, bacteria, fungi, and protist. So all types of pathogens have a simple life cycle. So what they do, they infect a host, they reproduce or they replicate if they're a virus, and then they spread from their host and then infect other organisms. And in plants, the pathogens, they kill or reduce the growth of many plants, which in turn will reduce the biodiversity on this planet. Plant pathogens of food crops can be very, very serious. In 1850, there was the Irish potato famine, and that caused deaths of large numbers of people through starvation because there was no food, as obviously potatoes are one of the staple diets. So one of the first plant diseases that I'd like to talk to you about is the tobacco mosaic virus. Now the tobacco mosaic virus infects tobacco, tomato and pepper plants, and it's transmitted by contact between plants, either naturally by insects or by the wind or on hands of farmers. And it infects the chloroplasts of the plant and leaves, and it changes their colour from green to yellow or white in a mosaic pattern, as you can see on the diagram on the right hand side. And it can also make the leaves crinkled and curl up. Now there's no cure at all for tobacco mosaic virus, so farmers' only option is to stop the crops from being infected or reduce the spread if they have the tobacco mosaic virus. So quick question now, and I'm going to give you a minute to write down your answer. So how does infected chloroplasts affect the plant? So I'm going to give you 1 minute to write down your answer. Off you go. Okay guys, so hopefully you have written down that if the chloroplast gets infected, it will reduce the plant's ability to photosynthesize. Remember, by photosynthesis, we produce glucose and make new proteins. Therefore, if the plant can't make new protein, it can't grow properly. So this can actually reduce farmers' crop yields. So the next type of plant disease that I'd like to talk to you about in a little bit more detail is rose black spot. And this is caused by a fungus which infects rose bushes. So what happens is it infects the leaves and it causes them to go black or purple. And then what usually happens is the rest of the leaves turn yellow and they can drop off the plant. So this reduces the plant's ability to photosynthesize and also reduces the growth. Now, black rose spot can be transmitted in air or in water, as well as direct contact by gardeners. So I would like you to write down for me how you think we can treat rose black spot. So I'm going to give you 30 seconds to write down your ideas. Off you go. Okay guys, so hopefully you've written down your answer to how is rose black spot treated. Because it is caused by a fungus, we can use fungicides to actually treat this disease, and also we can remove and destroy the infected leaves. So guys, I'd like now to talk to you about insects. So insects can infest plants as well as microbial pathogens. And a very common type of insect that actually invades plants are aphids. And these are found very commonly in this country, in our gardens and in our greenhouses. And what they do in particular is they infest roses and tomato plants. And they're usually found in large numbers on new shoots, in which they suck the sap out of the plant. Now what this does is it reduces the growth rate of the plant and eventually it will kill the plant. So how farmers or gardeners like to try and treat these is they actually try and cultivate ladybirds, because ladybirds eat aphids. So it's a very natural way of actually reducing them on tomato and rose bushes. So now I'd like to talk to you about mineral iron deficiencies and especially nitrates. So deficiency diseases in plants are caused by lack of certain vitamins or mineral ions, and plants naturally absorb nitrates in water through their roots. And nitrates are present in high levels in plant fertilizers. Now, without nitrates, a plant can't grow properly, which for a farmer, it reduces their crop yields. Now, thinking about nitrates, I would like you to tell me, how do plants use nitrates? So how do plants use nitrates? So I'm going to give you 30 seconds to write down your answer. Off you go. So hopefully you have written down, guys, how plants use nitrates, and what they do is they combine the nitrates with glucose to make amino acids. And then obviously amino acids are then used to make new proteins, and the protein then allows the plant to grow. Next mineral iron deficiency that I would like to talk to you about is magnesium. Now, plants use magnesium to make chlorophyll in their leaves. Now, chlorophyll is obviously the green pigment that is needed for photosynthesis. And plants that do not have enough magnesium suffer something called chlorosis, and they end up having yellow leaves. Okay, so an experiment that you can do to see the growth of plants in different nutrient solutions is as follows. So you can sow 10 barley seeds onto cotton wool in a petri dish, add 10 centimetres cubed of distilled water, seal the petri dish using tape to avoid any water loss or evaporation, and place it in a sunny windowsill to germinate, and then repeat this with 3 other petri dishes. Then about 1 week after germination, open up each petri dish, pour out any distilled water and add 10 centimetres cubed of the following solutions. So in petri dish 1, it will just be distilled water. In petri dish 2, it'll be a culture solution containing magnesium sulfate and potassium nitrate. In petri dish 3, there will be a culture solution containing magnesium sulfate, so this will be then deficient in nitrogen. Petri dish 4 will be a culture solution containing potassium nitrate, so they'll be deficient in magnesium. And then replace all petri dishes on the windowsill and leave for 2 weeks. Then after the 2 weeks is ended, open each petri dish and carefully remove each seedling from the cotton wool. And then what you're going to do is dry it in an oven and record the total dry mass of each set of 10 seedlings. So these are the results then obtained from this experiment. So petri dish 1, it was distilled water that was added, and the seedling dry mass was 2.1 grams. Petri dish 2 was a culture solution containing magnesium sulfate and potassium nitrate, and the seedling dry mass was 3.4 grams. Petri dish 3 was a culture solution containing magnesium sulfate, so this would be deficient then in nitrogen, and again the seedling dry mass is 2.3 grams. And then petri dish 4 is a culture solution containing potassium nitrate, so this will be then deficient in magnesium, and the seedling dry mass is 2.7 grams. So what I'd like you to do now, and I'm going to give you 3 minutes to answer this question, I would like you to tell me what conclusions that you can draw from this data. Now, when you are answering this, you must use the numbers in your answer. So it's a 3-mark question, so like I said, I'll give you 3 minutes. So 3 minutes to complete, off you go. Okay guys, so hopefully the conclusions that you've drawn from the data is that the average dry mass of the seedlings increases from 2.1 to 3.4 grams when water is replaced by a culture solution. So you get 1 mark for this. Then when the solution does not have nitrogen, the dry mass reduces to 2.3 grams and you get 1 mark for this. And then when the solution does not have magnesium, the dry mass reduces to 2.7 grams. So you get 1 mark also for that. And the reason is, as we know, that seedlings that are grown without nitrogen will be smaller than the others because we know that nitrogen is used to make proteins. So this is obviously the use then for growth and repair. And also seedlings that are grown without magnesium, these will appear to whoever did the experiment more yellow, and they will also be smaller because we know that magnesium is used to make chlorophyll, which is obviously very essential for photosynthesis. Okay, so what we need to talk about now is symptoms of plant diseases and how then we identify what pathogens actually cause the disease itself. So what I would like you to do is I would like you to list some symptoms that you may see on a plant which has a disease. So write down all the things that you might see that we've learnt about today, and also other things that might come into your head. So I'm going to give you a minute to list any symptoms that you might see on a plant that has disease. Off you go. Okay guys, so some symptoms that you might see on a plant which has disease is you might have seen that it's not grown very much. There may be spots on the leaves. There might be areas of decay on the plant, growths, some malformed stems, discoloration of parts of the plant, and also presence of pests on them. Okay, so now we need to talk about how can diseases in plants be identified. So if you've got a plant in your garden that you can see is diseased, how would you find out what pathogen is causing it or what insect is causing it? So I would like you to write down some ideas of what you would do to try and identify this. So I'm going to give you a minute to write down your ideas. Off you go. Okay guys, so these are some of the things that you can do. So you can make reference to a gardening manual, book or website. You can get laboratory analysis of testing kits which contain monoclonal antibodies, or you can take a cutting of the plant or a photo to a local garden centre, which they may then be able to help you identify what disease your plant has. Okay, so on this slide, we are now looking at the defences that a plant actually has to protect itself from actually getting some diseases. Now, they have three different types of defences: physical, chemical and mechanical. So the physical defences obviously prevent pathogens from infecting a plant, and the physical defences that they have may be bark, so that it's a layer of dead cells which forms a barrier, cellulose cell wall which acts as a barrier, and leaves have a waxy cuticle which also acts as a barrier. Chemical defences, so mint and witch hazel produce antibacterial chemicals that kill bacteria, and this is now often used in antiseptics for humans. And then some plants, such as stinging nettles, develop poisons to stop themselves from being eaten by herbivores, but please note that these do not defend plants from infections by pathogens. And then the next one, mechanical defences, these are defending from attack. So thorns and hairs on roses or cacti protect them from being eaten. Drooping leaves, so some plants close their leaves and point stems towards the ground when touched by an insect, so this makes it difficult for the insects to feed on their leaves. And then mimicry, so some plants mimic their surroundings, for example a pebble plant, to avoid being eaten by herbivores. Okay guys, so now we're going to do a quick quiz, so you're going to need your pen and paper ready. So I would like you to give 2 ways a disease can be identified in a plant. So 2 ways a disease can be identified in a plant. So I'm going to give you 30 seconds to write down your answers. Off you go. So hopefully you have written down that you can either make reference to a gardening manual, book or website. You can get a laboratory analysis of testing kits which contain monoclonal antibodies, or take a cutting of the plant or a photo to a local garden centre where they can help you identify your plant disease. So the next question is, give 1 physical defence response of a plant. So give 1 physical defence response of a plant. You've got 30 seconds. Off you go. Okay, so hopefully you have written down that a physical defence response of a plant, you could either have bark, which is a layer of dead cells which forms a barrier, a cellulose cell wall which acts as a barrier, or leaves have a waxy cuticle which also acts as a barrier. So next question, what happens if a plant doesn't get enough nitrates? So what happens if a plant doesn't get enough nitrates? So you have 30 seconds to write down your answer. Off you go. Okay guys, so hopefully you have written down that without nitrates, the plant can't make proteins and therefore it can't grow. Okay, so last question then guys. So give 2 mechanical adaptations of a plant for defense. So give 2 mechanical adaptations of a plant for defense. So I'm going to give you 30 seconds as always to write down your answers. Off you go. Okay, so you could have had 2 of any of the following: that thorns and hairs on plants such as roses or cacti protect them from being eaten. So with drooping leaves, so some plants close their leaves and points the stems towards the ground when touched by an insect, so this makes it difficult for the insect to feed on the leaves. Or by mimicry, so some plants mimic their surroundings, such as a pebble plant, to avoid being eaten by herbivores. Right guys, so now we've come to the end nearly of today's lesson, and I would like you to complete these summary notes as always. I'm going to give you 5 minutes to complete the notes. If you need any more time than that at the end, please pause the video. So today's summary: nitrates are needed to make protein so the plant can grow. A lack of nitrates causes stunted growth. Magnesium ions are needed for making chlorophyll, which is needed for photosynthesis. Without magnesium, plants suffer from chlorosis and have yellow leaves. Common signs that a plant has a disease are stunted growth, spots on the leaves, areas of decay, abnormal growths like lumps, and discoloration. To identify a plant disease, look up the signs in a gardening manual or on a gardening website. Send a sample to a laboratory for analysis. Plants' physical defences are the waxy cuticle, cell walls made from cellulose and layers of dead cells, e.g. bark on trees. These all act as a barrier to pathogens. Chemical defences: some plants produce antibacterial chemicals which kill bacteria, and some plants produce poisons to deter herbivores. And mechanical defences: some plants have thorns or hairs to stop animals from touching or eating them. Other plants have leaves which droop or curl when something touches them. Some plants mimic their surroundings. So you have 5 minutes to write down these notes, please guys. Like I said, if you need any more time, please pause the video. Off you go.